Okay, so today I'm going to speak to you about Aldolase's role in glycolysis and gluconeogenesis. Excuse my attire again, I just got done in the 110 degree weather coaching a swim practice, so I may look a little gross, but I'm still going to go for it. My name is Kristen James. I'm a student at Grand Canyon University. This is my final presentation for my class CHM 530, Biochemistry. So I'm going to go ahead and get started. So what is glycolysis? So glycolysis is a topic that we've been talking about for a little bit over the last week or so. Glycolysis is a series of reactions that extract energy from glucose. So it's taking the glucose energy, which is a sugar molecule, and pulling energy from it. It splits, how this occurs is it splits into two 3-carbon molecules. So it takes a molecule of glucose, it pulls it apart into 3 carbon molecules, 2 of them. Each one's actually called a pyruvate, and we'll go over. There are many, many more steps that go into that simple bullet point right there, but that's just kind of the overview of what occurs. And then it occurs in the cytosol, so you'll see here a cell. You have the plasma membrane, you have a bunch of organelles in here like the nucleus. And this area that kind of fills in all the rest of the areas around is called the cytosol, and that is where glycolysis occurs. It has been present for millions of years, so since it does not need oxygen, it is thought that it occurred on Earth even before we had oxygen in our atmosphere. So that's a very, very long time. The Earth is about 4.5 million years old, so the fact that it's been around for much longer than humans have been around is pretty impressive. And again, it is oxygen independent, so it does not need oxygen to function. Makes it very useful in a lot of situations and in a lot of organisms. And it's still present in almost all living organisms. Even though it doesn't need oxygen to happen, it does occur in animals that take in oxygen. And it is energy producing. Again, that's the main goal. We do glycolysis to get energy. And it results in a net of 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules. So altogether, we have the 2 ATP, the 2 NADH, which are the main molecules that produce energy and are derivatives of energy, while the pyruvate molecule is kind of the excess product that's left off. So there are kind of 2 versions of glycolysis when we're going over it. There's the novel version, which is the first version, and then the greatest hits version. So as we know, novels are long. They've got a lot of pages, they've got a lot of words, they've got a lot of text, they've got a lot of content. They're very beefed up. So this is technically the novel version of glycolysis. This is not even it, there's a whole nother slide. So to go through it kind of quick without focusing too much on it, since we're really focusing not on glycolysis as the main topic of this presentation. So what happens first is a phosphate group transfers from ATP to glucose. It's very highly reactive, so glucose 6-phosphate is created when this occurs, and now it's trapped inside the cell because it's too large. So when it's too big, it can't escape the cell, can't escape the cytoplasm through the plasma membrane anymore. It is trapped. So then that glucose 6-phosphate transferred into fructose 6-phosphate, which is an isomer. So it's a little bit different, but it's very similar in structure and shape. This fructose 6-phosphate is then produced and transferred into fructose 1,6-biphosphate, and it is produced from ATP transfer. So we go from that fructose 6-phosphate to biphosphate, with bi meaning 2, because ATP gives off a phosphate. So that happens by the phosphoructicanase enzyme, and this occurs and it speeds up that process or slows down the process. So if there's a lot of this around, this process occurs quicker than the opposite. But this biphosphate, so bi meaning 2-phosphate, is happening because of the ATP transfer. That P, the phosphate of the P, transfers over. Then coming down to the next step, fructose 1,6-biphosphate, it splits into 3 carbon sugars, and the results are all isomers of each other. So many steps throughout glycolysis produces products where they're very similar to the previous or original product, it's just slightly different. DHAP, converts glyceraldehyde to phosphate. These molecules then stay at equilibrium until they're used up. So DHAP and glyceraldehyde 2-phosphate are normally at equilibrium, so if one rises, the other will fall a little bit, it catches up. So it's almost like a scale, so instead of being tipped one side and tipped the other, it's more balanced right in the center. Stick with me. So continuing our novel version of glycolysis, that glycolyzed 3-phosphate is then oxidized to NAD+, and the NAD+ is reduced at the same time. So this occurs simultaneously within the cell, again in the cytoplasm. And then overall, these processes are exergonic. Okay, so they release energy, they give it off, and then 1,3-biphosphoglyceride is released. So that is what results from that step. So then going on, 1,3-biphosphoglyceride donates a phosphate group. So again, we were talking about originally accepting a phosphate group. Now we're giving off a phosphate group. So when that phosphate group is given off, ADP creates ATP, T meaning tri, so it's picking up a phosphate, which is then donated, and it turns this 1,3-biphosphoglyceride into 3-phosphoglyceride. So a lot of times when things are donated, you can tell by the name change in the prefix or suffix. But anyway, so then moving on, we got 2-phosphoglyceride forms that 3-phosphoglyceride with the ATP and ADP transfers. And then a molecule of water is lost to form 2-phosphoglyceride, resulting in PEP. Okay, so this is very important, and phosphenolpyruvate is another word for PEP, but it's short for PEP because obviously it's very, very hard to pronounce. It's got basically every letter in the alphabet in it, or at least it seems like it. So this molecule is unstable and it loses a phosphate group. So when that water is introduced or pulled away from the phosphoglyceride and that PEP forms, it's very unstable. Because of that, that's how we kind of end up getting our final product. So we need that instability to have our final product be created. So ADP accepts that phosphate group that has been lost. So ADP is just waiting, and all of a sudden it takes up this phosphate group and ATP is created. So what is left of the PEP minus that phosphate used to make ATP is called pyruvate, and that, like I said, is the end product left over from glycolysis. So we get the resulting ATP, but we also have the leftover or the garbage, kind of. So here's kind of a photo of all the steps without as much wording, more just going down the process. So we start out with molecules of glucose, we move to fructose, glycerolide and diphosphoglyceride, all the way down to our result, which is this pyruvate here. You can see we're producing our ATPs. So lots of words, very complicated, lots of small steps and sub-steps that go into glycolysis. And because of this, there is a greatest hits version of going over what exactly glycolysis is. And the greatest hits version separates glycolysis into two main chunks. And those two main chunks are the energy requiring phase and the energy releasing phase. So during the energy requiring phase, glucose gets rearranged. So as we talked about before, glucose is getting pulled apart, put together, pulled apart, put together. That occurs during the energy requiring phase. So energy in the form of ADP or ATP is required for these steps. Two phosphate groups attach, creating a modified sugar called fructose 1-biphosphate. So that results then in splitting that occurs, and it forms two phosphate-bearing 3-carbon sugars. And then finally, 2 ATP molecules are used up in total. So gets the name energy requiring from these 2 ATP molecules that are used up in the process. So that's first half, so part 1. Then followed by part 2, which is the energy releasing phase. So during this phase, energy is released or given off, which is the main goal of glycolysis. So each of those 3-ring carbon sugars is converted into pyruvate. So those 3-ring carbons from back before in the energy requiring phase are then made into pyruvate. And then 1 molecule of ATP and 1 molecule of NADH occur. They are produced. But because this happens twice, so this process occurs twice, therefore instead of having just 2 ATP and 2 NADH, we have 4 total net ATP and 2 total NADH. So it's a doubling. Instead of having just 2 and 1, we have 4 and 2 all together. That is our greatest hits version. We got a little bit of Whitney, got a little bit of greatest hits in the 1990s to help us remember that even though it's long and complicated, glycolysis has many, many steps, it is easy to kind of summarize the processes and then break it down from there. So focusing on one specific part of glycolysis is what we're going to do for the remainder of this presentation, and that is aldolase. So aldolase is a part of glycolysis. It is also known as fructose biphosphate aldolase. So here you'll see that. You'll see it right here. You'll see it over here. So aldolase right there. You can see it's present in skeletal muscle, heart, brain, and kidneys in humans. and other mammals and animals, and we're going to talk specifically about the different types and where they're located here in a bit. But mainly, in terms of location within the cell, our aldolase is located in the cytoplasm and the nucleus, so more central in the cell. Also, I skipped one. It catalyzes the reversible cleavage of fructose 1,6-diphosphate. So that's the main role it has. So if you think back to when we talked about the long list and even the shorter list, in that energy requiring phase, we had our fructose 1,6-phosphates and all their processing and maneuvering. So this is where this aldolase is going to come into play. Without it, it wouldn't really be able to happen, especially not at the speeds it needs to. So it's present in almost all cells. Almost every single cell in your body has this aldolase in it, but it's in very, very high levels in places like the liver and muscle and brain tissue. Okay, very, very important areas. It regulates cell contraction through reversible bingings. I definitely didn't mean that. The poolings of filaments. So it regulates how our cells contract, how they work, how they move. And at low levels of it, if we have a low level, so it causes disease, and we'll go specifically more into that in a second, but low levels of this aldolase represent skeletal muscle injury, which can either be due to a stress or a trauma or a lifelong disease that some may have from birth. So aldolase is known for breaking its CC bonds. So I know this may be a little bit hard to read, but I'm going to kind of go through it. I couldn't really get it much bigger. This is from our textbook. But this kind of goes through why aldolase is important. So it breaks those CC bonds. So some common reactions that form and break CC bonds are in biological systems, and these bonds and breakage have to occur with aldolase. So for both aldol condensations of the condensation, a carbon serves as a nucleophile, and the carbon of a carbonyl group serves as an electrophile. So you see that kind of shown up here, and it goes through the process showing the breakage of those carbon bonds, which wouldn't really be possible, and it wouldn't allow glycolysis to continue on if we didn't have that ability. So aldolases are not all the same. There are two different types. There are class 1 and class 2 enzymes. So class 1 enzymes are found in animals, animals like humans, and other mammals and vertebrates and all kinds of stuff like that. Any organism has these class 1 enzymes. They do not require metal ions, so we do not need metal ions to make them function properly. And they form a shift-base intermediate between a substrate carbonyl group and an active site lysine. So that is what's required for them, for them to be processed. And then class 2 enzymes are a little bit different. They are found in bacteria and they're found in fungi. They require an active site divalent metal ion. So although the ones in animals, class 1, do not need a metal they do not need a metal ion. The ones in class 2, they do require a metal ion. So I do have a list here of some of the metal ions that are used as divalent metal ions within the class 2 enzymes to help for functionality. Some of those are copper, nickel, cobalt, going down a little bit, calcium, zinc, you got lead, a very common one, along with magnesium. So these are just some examples of the different metals that help them function, but the basis comes in the difference between the two classes. So we focus more in biochemistry when we're studying humans on the class 1 enzymes, since that is what is in our bodies. So aldolase, even within the class 1 and class 2, they have many isoforms. What an isoform is a similar protein, has a similar functionality with similar amino acid sequences, but they are encoded by different genes or RNA transcripts. So although they may look similar and they may act similar, they're encoded in their DNA and the genes, and the RNA behind them is slightly different. So for each enzyme, such as aldolase, there are different types of the enzymes, so different isoforms. There are 3 specific ones with type 1 enzymes we're going to talk about that are found in the human body. The first one is A, and it's a glycotic enzyme in the majority of tissues. So this is what helps us a lot with completing glycolysis, as we were mentioning earlier. But then B is present in liver, small intestine, kidneys, and it converts fructose 1P into glycerolide. And that one is one that can cause a lot of diseases and problems in the human body if it's not functioning how it should. And then C is present in the brain, not quite in as much amounts, and it functions similar to the way A forms, as well as it functions similar to A and it forms similar to A. It really has a lot of the same reactions and processes that A does, So B is kind of the unique one out of A, B, and C aldolase isoforms. So aldolase also plays a role in gluconeogenesis. So gluconeogenesis, before we get into aldolase's role in it, it is the metabolic pathway that synthesizes glucose from non-sugars. So when we don't have glucose available, it's going to take in non-sugars, it's going to produce that sugar that we need to get our energy. So some examples are pyruvate, so the leftover process from glycolysis is actually then used for another process to hopefully get more glucose to produce more energy. Lactate's also another example that can be used. And it can occur in the liver or kidneys as well as aldolase's location, which is why they're kind of related to each other. And it supplies plasma glucose between meals. So if you think about your everyday, if you're like me, you probably have 2 to 3 main meals, but then lots of snacks in between the day, right? So lots of snacks here and there. The same thing happens with the cell. So even though it eats its main meals through glycolysis, the mass productions of energy, sometimes it needs a snack in between meals, and that's where this gluconeogenesis comes into play. So it's continuous in carnivores because they have a high energy demand, but in some animals and plants or in some animals and organisms, it's not quite as common. But in carnivores, we have it pretty much all the time going on because we need that constant energy to function the large bodies and the functionality that we have. So looking at aldolase, aldolase B plays a role in this carbohydrate metabolism. So if you see up here, you'll see glucose coming in, and we have over here our aldolase B and aldolase A helping to convert and make gluconeogenesis occur. And it catalyzes that F1P breakdown into glyceraldehyde and DHAP. So without it, 1, we couldn't make our main form of energy, and 2, we couldn't make our sub form of energy. So there has been some problems with aldolase. Obviously, everything in the body can mutate and go wrong, and just as everything else, aldolase can too. So I'm going to speak with you quick about aldolase A deficiency. I'm sorry, it's been a long day of talking, but aldolase deficiency plays a role in hereditary spherocytosis, so it's called HC for short. So here you'll see a picture of some people. This is going to represent this next point we're going to talk about. So autosomal recessive, it's metabolic disorder. So it is recessive. So it shows right here there are 2 carriers. So if a male and a female are carriers, meaning they do not show the disease, rather they carry it as a recessive trait. They have about a 25% chance of passing it on to their child who would be affected. Since it's autosomal recessive, you would need both copies from both parents of that recessive gene. They have a 50% chance of their offspring being carriers as well. So they would have the same status as the parents, being a carrier. They would not show the disease, they would not struggle and suffer from it, but instead they would carry it and they would then be able to pass it on to their youth and their children if they would have them. And then there's also a 25% chance of having no effect at all. You get all the clean genes, you wouldn't have to worry about it. So aldolase A deficiency can run in families. It is normally even somewhat rare in families that are affected because you only really have a 25% chance of being affected heavily, even though it is like 75% chance of at least having 1 gene of it. But aldolase A, it normally affects the blood cells. So it affects the blood cells and the muscle cells, which are those areas where it's more commonly found, there to help with contraction and movement. And some symptoms that a patient may experience if they have problems with their aldolase A levels and they have a deficiency, they may, the biggest thing they'll notice is red blood cells. So if they have blood taken, they'll notice a big change in their red blood cells. That's where the main indicator is going to be. They also may have a shortage of red blood cells, so this is known as anemia. Some people with anemia get originally diagnosed and they don't know that it's aldolase A deficiency, but that is a big indicator. You can also have yellowing of eyes and skin, so that's called jaundice. So if you have a yellowish tint normally noticed on the face or the yellowing of the whites of the eyes, that is another hint that someone may have an aldolase A deficiency. And one that is harder to kind of pick up on or harder to notice without medical procedures is a person with an aldolase A deficiency may have an enlarged spleen. So the main thing with this syndrome, so the big problem for patients who experience this and are diagnosed, is their red blood cells tend to act older than they are. So red blood cells, just like every part of the body and every cell in the body, has an age. They live and they die just like whole human organisms do. And unfortunately with aldolase A deficiency, these people or patients have red blood cells that act much older than they are. So even when they're newer and younger, they act old, causing them to die off earlier. So aldolase is a very, very important role. It plays a lot, very important part in making sure glycolysis and gluconeogenesis perform as they should, which supplies the body with energy in times of struggle and in times of pure sugars and the availability. So having that meal and that snack for the body energy-wise wouldn't really be possible without aldolase A, B, or C. So overall, it is very important to the body because we need our energy to survive. Some references I have up here, some came from some textbooks, some are from some scholarly journals. But I thank you, and if you have any questions, please feel free to email me at kjames@agufria.org. I'd be happy to answer any questions, and I'm looking forward to continuing in the class. Thank you.